Hi, students. Very good evening. So, before we start up with today's class, okay, so let me just launch a technical poll. So, please let me know if the audio, video, and the background is clear. No, you can't hear me well. Okay, let me check once. I'll relaunch again. It should be. Hello. Yeah, this should be okay. Again, I'm going to relaunch the poll. Please let me know if it's fine now. Is my voice clear to you? Yes. Thank you. So today we have to have a discussion or very important questions that appeared in 2023 till where we discussed. Okay, in the last class we discussed, I think, till the experiments that were conducted to understand uh, the search for genetic material, as well as we discussed about until DNA replication, I believe, correct? So we'll have a quick solving of around 15 questions which are related to what we discussed, and if the time permits we will discuss the rest of the topics further from where we left off. Is it okay? So it's all on you, so you have to answer today's question. So let's go. Here is our first question for the day. Yeah. Unequivocal proof of the DNA is a genetic material was first proposed by. So here are our first options actually: Wilkins and Franklin, Frederick Griffiths, Alfred Hershey and Martha Chase, and Avery McLean, McCarthy. So these are the scientists. Okay. So you have to tell which of the scientists provided the proof that DNA is a genetic material. Okay. So I'm going to launch a poll for you guys. Okay. So here's your poll. So please tell me, what do you think is the right answer? B. We have 1 minute. The rest of you, please try to answer this question. An unequivocal proof that DNA is a genetic material was first proposed by. All right. So let me end the poll. And 1 second. Yes. So we already have studied about the DNA as a search for the genetic material, and some of you have chosen the right option, and some of you have gone for, as usual, as expected, that is the Griffith, Frederick Griffith. But the right answer for this question is Hershey and Chase. Okay, that is Alfred Hershey and Martha Chase experiment. So these are the guys who actually proved that DNA is a genetic material. So see, when it comes to the search for genetic material, of course, Griffith performed one of the most remarkable experiments, that is the transformation principle. So, was Griffith able to tell locally that DNA was a genetic material? Definitely no, it was not possible. And even though Avery, McLean, and McCarthy performed the experiments on Griffiths and they searched the biochemical nature for what is the Griffiths experiment based on, and even then they could not conclude yeah, their experiment was 100% right and 100% was scientific, and it would be the ultimate one. but it did not convince 100% all the scientific community and did not shut all the critics. But Hershey and Chase experiment, that is the one that performed with bacteriophage and the E. coli bacteria, when they grow this label this bacteriophage with sulfur and phosphorus, radioactive sulfur and phosphorus. Ultimately, we understood that the cell had radioactive phosphorus and the supernatant did not have any radioactive phosphorus. So that proved that the DNA is a genetic material. So this is the right answer, and these are the scientists who just proved that DNA is a genetic material. All right. So it's all right. So here's your next question. Express sequence tags refers to. Okay. So here are your options. Certain important express gene, all the genes that is expressed as RNA, all genes that are expressed as proteins, all genes expressed or unexpressed. Okay. So I will launch the poll again. So here is your poll. It's okay. Just raise your opinion. If you don't know, anyways, we are gonna talk about it, right? Very nice. Very nice. Students from YouTube, please participate by raising your options or raising your opinion. Be very interactive. Okay. Come on, students. We have 40 seconds up, and we have more students to answer this. What are express sequence tags? Yes, very good. So let me end the poll, and we're out of time anyways. So you're absolutely right. Express sequence tags are any genes that are expressed as RNAs, and this question was asked in 2023 in NEET examinations. So usually what happens is whenever we are trying to locate or whenever we are trying to understand the expression of a gene, basically we go and target their RNAs. So RNAs are the ones that we look into so that we can just try to understand the sequence of the DNA. For example, if I have an mRNA sequence that is been synthesized, so I will be able to understand what might be the base pairs of a DNA, right? For example, if I have an mRNA sequence, okay. So on this mRNA is like this UU, GG, CC, AU, something like that. Then it is very easy for me to decode its DNA. So it will be like A, C, C, G, G, and T, and A, right? Something like this. So this would be the sequence of the gene that I would be looking for. So these sequences, whichever are expressed as mRNA, and they are called as express sequence tags. And basically, this helps us in locating, or this helps in understanding the sequence annotation of the gene that we're looking for. Okay, so you guys are absolutely right, and our correct option is option two. Okay, so let's go. What is the role of RNA polymerase 3 in the process of transcription in eukaryotes? So you have the following options, that is transcription of only snRNAs, transcription of rRNAs such as 28S, 18S, and 5.8S, and transcription of tRNA, 5S tRNA, and then snRNA, and then transcription of precursor mRNA. So I will launch the poll again. Okay, so let's see what's your answer. What is the role of RNA polymerase 3 in the process of transcription in eukaryotes? Yeah, good. Transcription of only snRNAs. Come on, students, you are just 20 seconds away. So give me your answer. Okay, we have this at D, transcription of precursor mRNA. Okay, so Before I answer this question, let me end the poll first. Students, I'm going to ask you a simple question. Is this topic being taught for you? So if yes, give me B. If no, give me C. If you have been taught this topic, yes means give me B. No means give me C. I think the poll is already there. Yeah. Wait, I will give you like this. Okay. If you know this topic, tell A. If you don't know this topic, tell B. Make it fast. Makes a lot of sense though. Okay. Okay. You know this topic, isn't it? So some of you are telling some, you know the topic. Okay. So that's very nice. But I am very surprised that you guys chose a very unusual answer. Okay. Anyways, so we will keep this here and I'm gonna just hold on, just share. Give me a second. Just give me a second. It'll make you very easy, something very easy. Okay. Mm, it'll make the things easy for you. Just give me a second. Okay. Yeah. Okay. Can you see this? Oh, hold on. Yeah. Can you see this, students? Can you see this slide? See, RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. So we have 3 RNA polymerase students and their location and their expression or whatever their function is very, very important. So our question is regarding RNA polymerase 3. I think you are able to see the slide. Let me if not, I will change it. Yes. I hope so. Okay. So let me get back. So I was telling you that and the role of RNA polymerase 3 is to synthesize 5s rRNA and then tRNA and snRNAs, that is small nuclear RNAs. So this is when it comes to RNA polymerase 1, whoever answered rRNA, it is strictly RNA polymerase 1 and it synthesizes all 13s and 28s RNA, sorry, rRNA are synthesized by our RNA polymerase 1. Whereas RNA polymerase 2, it synthesizes pre-mRNAs or mRNAs. Okay, so this is about it. Is it clear? Did you understand? Okay, one second. It transcribes 5s tRNA and 5s rRNA. So that should be your answer. Okay, where is my presentation? Yeah, this is my presentation, right? Yeah. Sure. Is it clear? Can we go for the next question? Okay. So here is your next question. And our next question is, give me a second. Yeah. Our next question is, in prokaryotes, you have the statements, two statements. First statement is, in prokaryotes, the positively charged DNA is held with some negatively charged proteins in a region called nucleoid. And you have the statement 2, in eukaryotes, the negatively charged DNA is wrapped around the positively charged histone octamer to form nucleosome. In the light of the above statements, choose the correct answer from the given option. So you have your option A, B, okay, C and D. So option A says statement 1 is incorrect, but statement 2 is true. And then you have the statement 1 and 2 are true. Statement 1 and 2 are false. Statement 1 is correct, but statement 2 is false. Okay. So I will launch the poll now. you just answer. Okay, sorry. Relaunch. So here is your poll. Okay, I'll just put it here. So tell, statement 1 says eukaryotes, the positively charged DNA is held with some negatively charged protein in a region called nucleoid. Statement 2 says, okay, statement 1 is incorrect, but statement 2 is correct. Okay, go on, keep answering. And then we have statement 2, that is in eukaryotes, the negatively charged DNA is wrapped around the positively charged histone of the bar to form nucleosome. Okay, so this is a statement 2. Okay. Students answer. Good, good, very nice, very nice. That means you guys have a very keen eye for picking up a very minute mistakes. I appreciate this. Okay, so let me end the poll. Your answers are absolutely right. Okay, so why? You already know that. So they have told that in prokaryotes, positively charged DNA, right? So we know that whether it is in eukaryotes or prokaryotes, the DNA is negatively charged. Okay, so if this was just negative, the rest of the statement is absolutely right. But since they have told it is positive, it is wrong. And then statement 2 is actually right. That is in eukaryotes, DNA is negatively charged, correct? Then it is wrapped around a positively charged histone optima. So that is also right. And it is called as nucleosome. That is also right. So that is why our statement 1 is incorrect and the statement 2 is true. I very much appreciate you students for this. This is very nice pick. Okay, so let's go for the next question. So we have the next question, that is, again, we have the two statements. The first statement is the process of copying genetic information from one strand of the DNA into the RNA is termed as transcription. And the statement 2 says a transcription unit in DNA is defined primarily by the three regions in the DNA. That is, the above statements choose the correct options given below. So this is your option A, B, and C, and D. Statement 1 is true, statement 2 is false, statement 1 is false, but statement 2 is true. Both statement 1 and 2 are true, both statement 1 and 2 are false. All right, so let me launch a poll. Yeah. So the process of copying genetic information from one strand of the DNA into RNA is termed as transcription. The transcriptional unit of the DNA is defined primarily by the three regions, that is promoter, the structural gene, and terminator. Yes, very good. Yeah, perfect. Very nice, very nice. Okay, so we have the good notes. I'll end the poll. Yeah. Guess what, students? You know what? We get the majority of the questions in 12th standard from this particular chapter. Very surprising, right? But it's very easy. And anyways, coming to this question, both statement 1 and 2 are true. That is what you have opted in your oaths, and absolutely right. So absolutely right. Why? That is a statement. So statement 1 says, what is transcription? Nothing but you have your DNA, which is a gene, and then you convert it into a messenger RNA. So that is nothing but transcription, which is what the statement 1 says. That is correct. And then the statement 2 says a transcription unit of the DNA is defined primarily by three regions, that is a promoter, structural gene, and a terminator. Again, these three regions are very must in any of the gene we talk about, that is having a promoter region and having a structural gene and a terminator region. So in both prokaryotes and eukaryotes, even though the promoters vary, the names of the promoters vary and the regions of the promoters vary. Promoters are conserved sequences, which we already know, and they actually help our RNA polymerase to come and attach to and start or initiate our transcription. It also, what do you tell? It also monitors, or it also matters the rate of, it also defines the rate of transcription. I can tell it also defines the rate of transcription. That is the second thing. And coming to the structural gene, this is where the mRNA is being transcribed, And you know the terminator region, whether it can be an inbuilt terminator or it can be a protein-based terminator sequences. So you guys are right. Without these three regions, there would be no gene, or the gene would be meaningless or non-functional, right? In simple terms. So you're right. So both statement 1 and 2 are true. Let's go for the next question. The scientists who conducted an experiment with P32 and S35, which are radioactive labeled, right? Labeled phages for demonstrating that DNA is a genetic material, are who? Okay. That is Watson and Crick. Then we have Hershey and Chase. Then we have Griffiths. Then we have Avery, McLean, and McCarthy. So here is your polls. It's a very easy question. After seeing your answers, I feel that it's way too simple for you right now. Yes, perfect. I'll wait for one more because we're getting consistent votes. Sorry. Absolutely right. Okay. So I'm ending the poll now. So, we already know that a famous Hershey and Chase experiment is the one where they performed experiment with radioactive phosphorus and radioactive sulfur to prove that DNA is a genetic material. So, and it becomes very obvious, right? So we know that what is the outcome of this experiment. By the end of this experiment, we see that there is a visibility of radioactive phosphorus in bacterial cell. Okay. So after the centrifugation, and this experiment is also called as Blender's experiment, right? We spoke about this in the last class. So in this Blender's experiment, what happens? The P32 will be found in the cells of the bacteria, which proves that the DNA is a genetic material. And the scientists who worked on it is Hershey and Chase. So you guys are making it very easy for me. So we have the next question. Statement 1 says RNA mutate at faster rate, and statement 2 says RNA can directly code for synthesis of proteins, hence can easily express the characters. In the light of the above statement, choose the correct option from the given below. Statement 1 is correct, statement 2 is false, statement 1 is correct, statement 2 is true. Statement 1 and 2 are true. What's the poll? Answer this. Wait, let me do this. Yeah. RNA being unstable, mutate faster. Mutate at faster rate. Second one, RNA directly code for synthesis of proteins. Hence, they can easily express the character. Which one do you think is correct? Okay. Do you have more options, more answers for this? Guys are absolutely right, and again, you chose the right option. That is, yes, RNA is very, very unstable. So the instability of RNA comes because of the chemical structure itself. We know that the RNA is more unstable because of the extra OH group that is present in the second position in its sugar group, right? So that is one thing. And we also know that it is a catalytic molecule and it mutates faster. And a very live example that we can get about this mutating faster is in viruses that have RNA as a genetic material. So that's why the statement 1 makes sense. Then RNA can directly code for the synthesis of proteins. Yeah, once we have the RNA can really code for the proteins. We don't need the help of DNA anymore because RNA is the one that carries the vital information that actually translates into the protein and can express into characteristics. So both the statements are just and both of the statements are right. So your option, what you have chosen, that is both statement 1 and 2 are true, is right. Okay. So let's go for the next question. As I say, we're making it very easy for me. Okay. So let me read the next question. The salient features of the genetic codes are the code is palindromic. Oh, okay. Yeah, the code is palindromic. UGA act as an initiator codon, and then the code is unambiguous and specific. The code is universal. Choose the appropriate answer from the following option given below. A and D only, D and C only, A and B only, and C and D only. So I'll launch this poll again. Go for your right options. Okay, I will just give this A. This is A, this is B, this is C, this is D. Okay. So fast. That is C and D. Very nice. I don't know which students are answering, but I like you guys. Very fast, and you're picking up points so nicely. You're not thinking. You're not giving your mind. You're like very natural. I'm not buttering you, but it's true. I'm happy to see the responses like this. Especially, I'm not solving an average question. It's a PYQs, that is previous year questions. All right. So come on. I'm waiting for one more response. There's other response. All right. So let me stop this poll. Yes, let's see. So according to the question, so if you look at the question, what do we have, students, here? We know what is a genetic code. The code is triplet. So we know that we have genetic codes. And if you don't know what is genetic code, let me break it down for you. For everything that is present in our DNA, right? For example, let's say our DNA is made up of, let us say, these are the possible nucleotides, that is ATCG. And you can see that ATCG nucleotides can be arranged in any fashion. Okay, like this, for example, I can get CCGG as well. So this is what is present in our DNA. Now, if I convert this into a messenger RNA, so this messenger RNA will be U, then GG, then again CC, then again AA, then again GG or CC, something like this. So this is my messenger RNA. So what happens after creating a messenger RNA? So our body or the biology is so smart that it has created codes for what has to be assembled, for what codes it has. For example, we can just divide these codes into groups of 3. So UG will code for one amino acid. Similarly, GCC will code for one amino acid. Similarly, AAG will code for another amino acid. So for every 3 codes, you have one different amino acids. So basically, we have around 64 codes, okay, out of which 61 codes for amino acids, and there are only 20 amino acids. So that will actually naturally state the properties of our genetic code. That is, the code is triplet. That means always there are codes come in 1, 2, 3. Okay, triplets. Next one, code is universal. Code is universal in the sense, see, we have something like code is universal. Whether you see a bacteria or a human being or any other organisms that you come across, if AUG codes for methionine in bacteria, the same AUG codes for methionine even in human beings. So after all, you are not so special in this universe, right? In this living universe. So that means all of the living organisms, if they have DNA, so they have to prepare proteins, then that means all of them follow the same genetic code. Very natural. Then 3 codes, such as I call them WA, WAG, and UGA. So this is my way of remembering. So you might call this as UAA, UAG, and UGA. But I have a sound that makes me easily remember. It makes it easy for me to remember. So all of these three codes, they are called as stop codons because they do not code for any amino acid. Okay, so then you have something called as AUG, which is also a start codon or initiator codon, or it is also called as a methionine. Okay, so we call it as methionine coding code. That is, AUG is also called as methionine code, or it is also called as an initiator codon. So there is a very important aspect that you have to understand here. What is that? especially when it comes to prokaryotes. Listen to me so that it will help you. So especially when it comes to prokaryotes and when it comes to eukaryotes, when we talk about translation, people. So when we talk about translation, so you know that in eukaryotes, we have something called as 5' capping. You know that, right? When we talk about RNA editing, we have capping, tailing, and we have something called as splicing. When we talk about 5' capping, we attach a 7-methyl guanosine cap, right? 7-methyl guanosine cap, correct, right? And here in prokaryotes, this cap is not attached, correct, right? So what is happening here now in prokaryotes, if you see, their mRNA has an inbuilt AUG sequence at the very beginning of the 5' end, okay, of the mRNA. So now what happens during the translation, like during the protein construction, the first tRNA, correct, right? The first tRNA, which brings this AUG, should bring what kind of an amino acid? It has to bring methionine amino acid, right? If there is a code called as AUG, then definitely it should bring methionine. But the type of methionine that comes very beginning of the translation. So it's the first amino acid that has to be added, isn't it? So the tRNA that brings a methionine is not a normal methionine. So this initiator methionine is something called as formylated methionine. So why do we call it as formylated methionine? Because a formalin group is added to this methionine. So that is why what makes it an initiator methionine. Understood? So the tRNA is also called as MET F tRNA. So this MET F tRNA, that is methionine formylated tRNA, is also called as initiator tRNA, and this happens exclusively with respect to our prokaryotes. I hope you understood this. AUG in prokaryotes is an initiator codon. Absolutely right. Why? Because a specific type of amino acid called as formylated methionine is added at the very beginning of the translation. That is why it has a significance of initiation, dual function. When it comes to eukaryotes, does it happen the same? Definitely no. It does not happen the same. Why it does not happen the same? If you look at the eukaryotes, in eukaryotes, we already have something called as MET-F cap, that is 7-methionine methyl gynosine cap. So this 7-methyl gynosine cap helps in the initiation process, and formylated methionine is not added in eukaryotes, but it is added exclusively in the prokaryotes. I hope this information helps you guys. Okay. Yes. And the code is unambiguous. The code is unambiguous means, excuse me, now it does not have two different meanings. If one code, hold on. What happened? Okay, now the response. Did you understand? So the code is unambiguous means it does not have a different meaning. So if I call AUG here is for methionine, even in other countries it is AUG only, that is the same meaning. So there is no dual meaning for a particular sentence. That is what is called as unambiguous. Now, the code do not have any punctuation. So whenever we write a genetic code, like how I have split it into three groups, we don't do that. Once we start from the 5' end, we will all go till the end of 3' end, and we don't put any comma or punctuations or full stops or exclamatory marks, anything, right? So this is about genetic codes, and what options you chose, that is the C and D, are absolutely right. You're correct. That is, code is unambiguous and specific, true, and code is universal, correct, and code is degenerate. What is code is degenerate? Right? So two codes can code for same amino acids. What is two codes can code for same amino acids? So if you look at your textbook, if I have something called as UUC and the middle, like UAC and then UGC, and then you have your let's say UCC. So if you can see this, they will code for the same amino acid. Okay, so you just check it out. Whatever the middle terms, you can see they have only the middle codes are changed. So if you see the groups of these, you can see that this is a different code and this is a different code, but they will be coding for the same amino acid. So this is called as a degenerate nature of the genetic code. So these are the features of our genetic code, I think, which you're already aware, which is given in the textbook. And one specific amino code, that is AUG. Why it is so special, especially in prokaryotes, I just explained you. If you have any doubts, you can let me know. Okay, let's go for the next question. Read the following statements and choose the set of correct statements here. Yeah, you have the first statement, that is euchromatin is loosely packed chromatin. Heterochromatin is transcriptionally active. Histone octamer is wrapped by negatively charged DNA in nucleosome. Histone are rich in lysine and arginine. Typically, nucleosome consists of 400 base pairs of DNA helix. So correct, choose the correct answers from the given option. So your options are A, C, E, N only, B, D, E, N only, and then A, C, D only, and then B, B, E only. Okay. So I will relaunch the poll. Okay, you have the poll, students. Let's go. Do you guys have some cheat code? I took longer time to read it than you answered. Okay, we have for 20 more seconds. Make it fast. Good. So the options are, I can see. Okay, so let me end the poll now. Let's see what you guys got. I think you're all sitting in the same class and discussing. May probably, that's the possibilities. Let's see. So you have A, C, and D. So majority says C is the right option. Let's see. Euchromatin is loosely packed chromatin. Very, very true. So last class we discussed about packing of DNA, and I told that euchromatin is loosely packed, and this is where the transcriptionally active DNA is present. When it comes to B, heterochromatin is transcriptionally active. I'm sorry. The network issue, a small issue. Guys, can you make me a host for a second? Who is the host now? Gandhi Nagar, JNV Gandhi Nagar, please. Yeah, reclaim host. So I'm host. Yeah. Sorry, there was network issue. Yeah. Yeah. So coming to the third option, that is typically nucleosome consists of 400 base pairs. So this is wrong. Around 150 to 200 base pairs of DNA is wrapped around a double helix. So I think your textbook says 200 base pairs, and usually it is 150 base pairs. So your option for 3, that is A, B, C only. So that is the right option. You guys are correct. Okay. So let's go for the next question. Okay. So we have something? No. DNA polymorphs form the basis of. So you have the following option, that is genetic mapping, DNA fingerprinting, both genetic mapping and DNA fingerprinting. So what is your option? So you have your polls. Hold on. Yeah. So I think the polls Hold on. Please tell me. DNA polymorphs forms the basis of translation, genetic mapping, DNA fingerprinting, both. Very good, very good. True. Now we have controversy. Okay. So let me end the poll and I'll discuss this controversy with you. DNA fingerprinting, and some of you have chose both. What is polymorphic DNA? What happens is whenever we see lots of genes, right? So we have around 25,000 to 30,000 genes on an estimate. So that is what the scientific data says. Now, if I just compare my genes into another human being, 99.5% of it is similar. Very exact genes will be there. So we can't even differentiate what's the difference between me and him. There is no way you can find out from the expressing DNA that this is the difference between this guy and this guy, because all the proteins are same in all human beings. But what is something different? The something that sets us different is a something that do not express, that is called as polymorphs. And this polymorphic DNA, actually what happens is from one generation to another generation, you can see that this repetitive sequence, which we call it as VNTRs or STRs. So this VNTRs, that is variable number of tandem repeats or short tandem repeats. Okay. So what happens is they are highly mutative forms of DNA, which are present in mini satellite or microsatellite regions of our chromosomes. So what happens whenever, let's say that my dad has a repetitive sequence of 80 rich region as a VNTR variable number of standard repeats. Let's say he has around 250 base pairs of repetitive units. 250 repetitions are there. Now, myself, I might have around 75. There is no correlation between that. You understand my point? It is very unique for them. When we talk about fingerprinting, so we talk about the uniqueness of a person. So that uniqueness comes with the number of repeats, and that is only restricted to me, not even my dad, not even my mom, not even my grandparents, and it doesn't occur in any of the ancestors of human beings. So that is only possible in a polymorphic DNA from one generation to another generation. So this tandem repeats keep changing in a very variability. High variability is there. So there is no predictability. You can't predict this will be the number of tandem repeats for the next generation. So that is why the Alex Jeffrey picked out this mapping, which DNA belongs to which human being. So that is how the DNA fingerprinting comes. But when it comes to genetic mapping, we go with something called as expressing RNAs. We initially talked about express sequence tags, right? So where we target the gene that is expressing. So VNTRs will not benefit us in going for gene mapping. VNTRs helping in DNA fingerprinting. That is why we have one of the greatest application, that is DNA fingerprinting, not genetic mapping. Clear? Okay. Next question. Excuse me. Which of the following is not criteria of genetic material? So should not provide the scope for the changes of evolution. Should be able to express itself in the form of Mendelian characters. Should be able to generate its replica. Should be stable chemically and structurally. Okay, I'll relaunch the poll. Chalo. Very easy question. Which of the following is not a criteria for genetic material? Correct. I was surprised. You took a little bit of time. Actually, you're doing it so fast. Hello. Okay, I'll end the poll. Yes, your answer that is fusion. So that is a wrong option, right? So the very important characteristic feature of the genetic material is that it should provide the scope for the evolution. So that's a very, very important criteria. Correct. So that is why our option A is correct, but the given statement is wrong. If the gene do not have the character for the showing evolution and recombination, then there is no possibility of the life to survive with the changing environment and the changing climate, or whatever the changes this world offers in our living, as a, in the biodiversity. It's not possible to cope up. So evolution is something that makes organism to survive irrespective of what comes next, right? So next one. Which of the following experiments of Frederick Griffith resulted in the discovery of bacterial transformation? It's an interesting question, even though it is easy. Okay, so let's see. So here is your question. Yeah. Which of the following experiments of Frederick Griffith resulted in the discovery? He takes the estrine heat killed, injects into the mice lived. It takes the estrine heat killed, plus he takes the R strain living, injects into the mice, mice died. Estrine injected into the mice, mice died. R strain injected into the mice, mice lived. So which of this following led to the transformation experiment? I mean, like, resulted in discovery of bacterial transformation. Yes, perfect. The answer is right. That is when he takes the heat killed estrine and then he takes the R strain and then injects into the mice died. Actually, we discussed about this, right? So we discussed about what is transformation and how the conjugation comes into this play, etc. So you're right. Your option 2 is absolutely correct. So the next one. Wait 1 second. Yeah. Your next question says, with respect to nucleosome, which of the following statement is incorrect? Nucleosome contains around 120 base pairs of DNA helix. Nucleosome are seen as beads on the string under the electron microscope. DNA is wrapped around positively charged histone octamer to form nucleosome. And nucleosome is a repetitive unit of a chromatin. Okay, which of the following statement is incorrect? Absolutely right. Right? Your answer, that is nucleosome contains around 120 base pairs of DNA helix, is the wrong one. Now give me the right option. How many of the base pairs are wrapped around the nucleosome in a histone octamer? Type your answer. How many base pairs of DNA helix is wrapped around a histone octamer? Did you get the question? The question is, yeah, you chose the right option. The right option is nucleosome contains 120 base pairs of DNA helix, which is wrong. Now, what is the right? How many base pairs? How many base pairs of DNA is wound around a DNA helix? Okay, should I give you options? I'll give you an option. Okay, options are, okay, options are 150 base pairs, 200 base pairs, 250 base pairs, and 300 base pairs. So you have here A, B, C, D. Come on, I will make this easy for you. Okay, so go for it. 200 base pairs. That is what your textbook says. Absolutely true. Correct. So each of the histone octamer can wind around 200 base pairs of the DNA helix. So that is what makes them and nucleosome, like the beats on the string model. But the rest of the options is absolutely right. All these three options. So the option what you chose, option one, is wrong. So let's next go for the next question. If the DNA contained sulfur, it's an interesting question. Okay. If the DNA contained sulfur instead of phosphorus and protein contained phosphorus instead of sulfur, what would have been the outcome of the Hershey and Chase experiment? No radioactive sulfur in bacterial cell, both radioactive sulfur and phosphorus in bacterial cell, radioactive sulfurs in bacterial cell, radioactive phosphorus in bacterial cell. So let's go. What do you think? If DNA contains sulfur, no radioactive sulfur in bacteria. Are you sure? Read the question. If the DNA contained sulfur instead of phosphorus and protein contained phosphorus instead of sulfur, what would have been the outcome of Hershey and Chase experiment? Okay, so I will end the poll. Now let us analyze. According to Hershey and Chase experiment, what happened? When we had radioactive phosphorus, that is P32, labeled bacteriophage when it infected, in the supernatant, we had supernatant and the pellet, right? When we did the blending and centrifugation. So pellet had radioactive phosphorus, right? Now what happened? This radioactive phosphorus got into the bacterial cell because the phosphorus was present in the DNA of the bacteriophage, and that got injected into the bacterial cell, and the blending happened, and the pellet contained both bacterial cell and the DNA. That's what made this bacterial cell radioactive. Now it is interchanged. The sulfur is placed instead of phosphorus, and the phosphorus is placed instead of sulfur. Now, you tell me, radioactive sulfur will be found in bacterial cells or not? What do you think? Do you agree or not? Again, I'm going to launch the poll. Now you choose your right option. What do you think is the right option now? If the DNA contains sulfur instead of phosphorus and protein sulfur, what would have happened? What is the outcome of experiment? The outcome would be found because when the phosphorus in bacteria was found, correct? Obviously, the radioactive sulfur gets into the bacterial cell through bacteriophage, which is a genetic material, which is the DNA. Ultimately, whichever the genetic material is transferred, that is what should appear in the bacterial cell. So your option 3 is wrong, not the option 1. Okay. So I will end the poll. Let's go for the next question. So the final question probably, let's try. Yeah. Against the codon, read the see the question carefully. Very interesting question again. Against the codon 5' UAC 3'. Next, what would be the sequence of anticodon on tRNA? So you have your options: 5' AUG 3', then you have ATG, then you have GTA, then you have GUA, and this is 3'. Okay. So try to answer this. Very, very interesting question. Yeah. I think the poll is on. If you have a codon 5' UAC, what should be the anticodon on the tRNA? This question was asked last year in exam. Oh no. You had a very sharp eye, okay? But I will give you one more chance to reconsider. You can choose twice. Choose another option. Yeah, tell me. I'm here. Hi. See, the point is you have to check the direction also, right? Direction is very, very important. So if I have 5 prime U, A, C, and then 3 prime, then my sequence will have to go from, see, what should you become what, you will become A, and the A will become T, and am I right? A will become, sorry, no T, AU, correct, AU, and C will become G. So here I have to consider the orientation of this. So obviously the only option, so this should be the answer, correct? So what did you do? What happened? So GUA, it comes to genetics. So your option should be 5 prime GUA. Did you? I think, I hope you understood, right? Awesome. So you got majority of the answers correct. So which was very, very nice. So thank you very much for attending the session. So here we end. All right. So I'm going to end the poll. So I'll see you in tomorrow's class and we'll solve more questions on the further topics. So be prepared for the tomorrow's class and read the following topics which